So good morning everybody. So we will continue our discussion on translation of proteins. Okay. So when we see about the translation, is basically translation is DNA replication is like printing a copy of all the pages from the book that we have already discussed. Like you are ultimately decoding the total sequence of DNA in replication, right? In transcription, only certain areas of the DNAs are copied, like copying a page from the book. That is like Xerox copy of a particular page of a book. So this is what is. Now when we talk about translation, it's genetic information of the DNA is transcribed or copied to the messenger RNA, and then messenger is translated to functional protein. So ultimately now what is happening, the DNA sequence which is already there decoded to form mRNA, we are going to translate the message or the information which is in the mRNA in the form of amino acid when we read the sequence of mRNA triplet codons. Okay. So during transcription what happens, the message of DNA is copied into a language of nucleotides, that is 4-letter language. Now during translation, this mRNA 4-letter language is translated into an amino acid which is 20-letter language. Okay. So this is what the translation is. So we are ultimately decoding the mRNA sequence to form a protein through by reading the frame sequence of triplet codons. Now this is what the DNA strands look like. It is having a 5' to 3' end, and this one is the 3' to 5' end. Okay. So 5' to 3' you have adenine to thymine, this is the thing. Now when we decode, we are ultimately, the sequence which we are having in mRNA is similar to that of non-template strand, if you observe, because we are copying from the template strand. If thymine, then adenine; if cytosine, then guanine; if guanine, then cytosine; if adenine, then it should be thymine, but thymine is not present in RNA, so it should be uracil. Then adenine, uracil, thymine is present in the DNA. This will give rise to adenine. So what basically we see in mRNA, we ultimately, the sequence of the mRNA will be similar to that of the template strand. Sorry, similar, it should not be similar to that of template strand, but similar to that of non-template strand. Okay. So this ultimately forms a sequence of a protein which an amino acid differentiated from amino acid. Now this is what we are going to learn in today's class. Now when we talk about translation, there are basic features which we know about it. One is the transfer RNA, the second one we should know about mRNA and the genetic code, which is extremely important. The third is ribosomal assembly, and the fourth is translation process. So with this process we continue our discussion on translation of proteins. So when we talk about translation, the first thing which comes in our mind is tRNA. tRNA is nothing but the adapter molecule which adapts the message from the mRNA for the protein synthesis. So in the tRNA, if a short question is asked, short note sometimes it appear in the exam about the tRNA, this is nothing but a clover leaf appearance. And what we notice here, the 3' end is having ACC sequence, which is extremely little lengthier, longer than the 5' end. Okay, that's the first thing you're going to note. And this is the place at the 3' end only the amino acid will go and bind. Okay, so amino acid will, when it get activated, there is something called the activation of tRNA. The moment tRNA gets activated to start the synthesis of proteins, at the 3' end you see the amino acids are usually bound. Now this sequence in which the amino acids are bound, which will determined by the anticodon sequence in this loop. Okay. So basically there is a 3' sequence which is little lengthier, then you have a dihydrouridine loop, okay, dihydrouridine loop, then you have a pseudouridine loop this side, and you have short extra arm. Normally it is have 95 nucleotides in this mRNA, in this tRNA. Okay. And there is something called anticodon loop. This is the anticodon loop. Okay, this is anticodon loop. Now we are going to read this anticodon loop with the codons. The codons are nothing but the 4-letter languages which are present in the mRNA. Okay. Now anticodon arms of the tRNA recognizes codons in the mRNA. Specificity of the tRNA residues in the anticodon area. Okay, specificity of the tRNA residues in the anticodon area, and anticodon has a base sequence complementary to that of codons in the mRNA. Okay? Anticodons has base sequence complementary to that of the codons of mRNA. You see here, suppose here inosine is there, guanine is there, cytosine is there. So, here you have a complementary sequence in the codons. These complementary sequences in the codons will determine what exactly tRNA should fit with this. Suppose you have AGU, suppose AGU is coding for phenylalanine, okay? AGU is coding for phenylalanine, suppose. Now, when it codes for phenylalanine, what do you expect to have? You expect to have in the anticodon arm complementary to this sequence, because when it is complementary, then only it goes and bind to this sequence which is coding for phenylalanine. Now, this complementary sequence will determine the binding of this amino acid in the 3' end. Okay? So, 3' end binding will be determined by the sequence of anticodon, the nucleotide sequence which is present in the anticodon loop, and that will be further determined by the codon in the mRNA. Okay? Now, here you see, as we discussed, 3' end, this is a 5' end, this is pseudoadenine loop, this is dihydroxyuridine loop, this is short extra arm, and this is the anticodon loop. Now, you see here, this is what we are talking. These triplet codons are red, okay, these triplet codons are red, and these triplet codons will be complementary to that of anticodon, and this anticodon loop will be determined where exactly this codon will be red, and accordingly this attachment of amino acids, different variation of amino acid will happen. So, ultimately you see the tRNA will carry around 20 amino acids, because these are the functional amino acids which will help you in the protein synthesis. So, these 20 amino acids will be carried by these tRNAs, and this tRNA carrying capacity of this amino acid will determine on the sequence of codons. Okay? So, GAG suppose is coding for tyrosine, suppose. Okay? So, when GAG is coding for tyrosine, you should observe that in the anticodon arm of the tRNA, these nucleotide will be just complementary. Okay, this will be complementary so that they can go and bind. And when it binds, why it has to bind? Because then only the amino acid will be present, and that will be consecutively added up with several amino acids to form a peptide linkage and further giving rise to protein. We will see that part. Now, you see here, suppose tyrosine is coded by these 3 amino codons. Now, that particular tRNA will go and bind to this codon who has the sequence of nucleotide in the anticodon loop complementary to that of the sequence which is coding for tyrosine in mRNA. Okay? So, these are the reading frames. 3 nucleotide is a reading frame, and these are considered as codon. Now, you see one thing here, when we read this frame, there is no overlap. Suppose this is one frame, this one frame, this one, this one, this one. You cannot see, you leave one and you start reading from here, In that case, what happens? The total amino acid sequence will also alter in the protein if you mislead or misread one particular nucleotide while reading these frames from mRNA. So, that is why reading these codons in the form of triplet in this mRNA is very important. That will only determine the sequence of amino acid being bound for forming a protein. Okay? Now, this is what we have discussed earlier also. Now, you see here, this alanine, one amino acid, it is being coded by several codons. Arginine, coded by several codons, you see, AGA, AGG, CAG, CGG, CGU. Aspartic acid, coded by several codons. So, maximum what you observe, one particular amino acid is coded maximum by 6 codons. Okay, 6 codons. Methionine, you see, have only one codon, AUG. AUG is a start codon, initiation codon. Okay. Similarly, tryptophan, you have UGG, only one codon. So, maximum you have 6 codons, minimum you have 1 codon. And these 3 nucleotide sequence in the mRNA in the form of UAA, UAG and UGA. These are what? These are nothing but stop codon. These are nothing but stop codon. Okay. Now, in the latest thing, we have seen that there is something called as amino acid like pyrrolysine. Okay, and selenocysteine. These two are new amino acid joined. These will be coded by UAG and UGA. Okay, so let's consider this, and then we'll find that only one particular stop codon is currently available, because this UAG and UGA is coding for pyrrolysine and selenocysteine amino acid. These are added up, 21 and 22nd amino acid. Okay. So this is what we are going to see. So anticodons 3' to 5' sequence that matches complementary to 5' 3. This is what sequence? This is a 3' end. Okay, this is a 3' end. So when this is a 3' end, obviously this will be 5' end, 5 to 3'. This is 5' end. So this will be 3' and this will be 5'. So the sequence of anticodon loop will be complementary to that of 5' to 3' sequence. Okay. Acceptor arm is the one which has amino acid the 3' end. This is nothing but additional, and this is the amino acid. So this is considered as acceptor arm. This is acceptor arm for amino acid. Okay, this is acceptor arm for amino acid. Now T and D loop. This is the T loop and dihydrourine loop. This provide structure for interface with amino acid tRNA synthetase. Why I am telling you? Because this enzyme amino acid tRNA synthetase is an important enzyme, and this amino acid tRNA synthetase will be used to activate this tRNA, which will be used to activate this tRNA. Okay. So for activation of tRNA, you need amino acid tRNA. So these two loops which are present, this will help the enzyme which activates the tRNA to bind. So tRNA can fit to the enzyme exactly. So that's why these T and D loops of the tRNA will provide an interface so that amino acid tRNA synthetase can go and bind, and activation of tRNA takes place. Now what happens, the activation of tRNA will take place only that moment of time in cytosol when all the free tRNAs are present. When you have seen that mRNA has been decoded from the DNA, and from heteronuclear mRNA it comes to a normal mRNA, which is a matured mRNA, and that matured mRNA comes from nucleosomes to the cytoplasm. So when the mRNA comes to cytoplasm, it's understood now we have to decode the message from the DNA to the protein via mRNA. So that moment of time only you expect the free tRNAs are ready, and they are getting activated by amino acid tRNA synthetase and bind with the tRNA. Okay. So that they recognize this anticodons. The anticodons which is present in the tRNA will recognize the codons in the mRNA sequence. You get my point? So mRNA sequence, which has to be read in the form of triple nucleotides, that will be complementary to that of the anticodon loop which is present in tRNA. And remember one thing, it's not all the tRNA will go and bind to the same RNA. The sequence of these codons which are present in mRNA will determine which exact tRNA should go and bind. So for that, the sequence in the anticodon loops should be always see that it's complementary to that of mRNA. Okay? So this part you have to very nicely understood. So first is activation. Once the activation is happening, we have to see which particular amino acid this tRNA is taking. The amino acid binding to the acceptor arm of the tRNA will be determined by the sequence of anticodons which is present, the nucleotides. and these nucleotides will be nothing but complementary to that of the amino acid which is coding. So if it is coding for phenylalanine, suppose U is coding for phenylalanine, in this anticodon loop it should be complementary to that, it should be G. Then only it will code for phenylalanine, or then it will carry phenylalanine. Is that part is clear? So what we see here, there are several codons coding for one amino acid. But have you ever observed that several amino acids are coded by a single codon? No. So what we observe there, we observe that suppose there are 6 codons maximum, and you have amino acid 1, amino acid 2, amino acid 4, 5, 6. Okay? So these different codons will code for the same amino acid. Okay? But one amino acid or several amino acids will not be coded by one codon. Part is clear? So several codons will code for amino acid, but several amino acids will not be coded by single codon. Okay? That part you please make it clear. Now when we talk about the genetic code, we observe that these are read in the form of triplet codons, and these are universal in nature. Suppose you have a genetic code coding for any amino acid for human being, the same will be coding for any other species of the earth. It is not that this is only for humans, for earthworm is different, for porcine is different, for giraffe is different, for cow is different. No, it is universal. The third part is degeneracy, because there are several codons coding for one amino acid. So there comes a degeneracy. Then unambiguous, it's also unambiguous. Then non-overlapping, no punctuation. So these are the characteristic feature of genetic codes which are coding for amino acid. We will see one by one these properties. So first is we talk about triplet codons. In triplet codons what happens? These codons are mRNA codon. Each codon is consecutive sequence of the 3 bases on the mRNA. UUU coding for phenylalanine, AUG coding for methionine. Okay? So these triplet codons are specific. The second feature is non-overlapping. Non-overlapping. When we talk about non-overlapping, these codons are consecutive. These codons are consecutive. Therefore, the starting point is extremely important. As I told you, if you have these many sequences, suppose you start from here, you have these triplet codons. Okay? Now you miss one nucleotide and start from here. Then what happens you see? The total information which is there is totally altered. That's what you see in case of hemoglobinopathies, okay? Sickle cell anemia, all these missense mutations, all this happens, single base mutations, okay? This is all due to, if you read the reading frames in a different way. So this is important here. The codons are consecutive, so it has to be read in the form of triplet codon. You cannot just miss one and start reading UGC, AUC, because AUC will be coding for totally different amino acid. So you will have a different protein which will be nonsense, or protein which is non-useful. And I am telling you, when we synthesize proteins from this mRNA sequence, the heavy resources of energy is required in the form of ATP. So we cannot just tend to waste our ATP for this synthesizing ridiculous nonsense protein. So that's why this is what the non-overlapping means. We have to see, we have to read these codons in the continuous manner. This is important. Okay? The third is no punctuation. You cannot put a break, you cannot put a full stop, you cannot put exclamation, no comma, no semicolon. When you start, it should be consecutive and continuous. Okay? So AUG, CUG, CUA, like this, it keeps moving. It's not that you stop here and again start from here. No. When you read the frames, it should be continuous and non-stop. You keep reading. And when you have termination, you can stop here. That is what the non-punctuation means. When we talk about degeneracy, it is degeneracy there are 61 codes which is coding for 20 amino acids. Why? Because total of triplet codons is 64 amino acids, isn't it? Of the 64, we have removed 3 amino acids because there are stop codons, isn't it? So you have 61 amino acid codons which are coding for 20 amino acids, because there are 20 tRNAs which will be taking up this amino acid. So one amino acid has more than one codon. This is what the degeneracy means. Serine has 6 codons, glycine has 4 codons. This is what the degeneracy of codons means, the fourth property. So this is clear. Now this one is nothing but vowel positioning. This is another important area where Vowels hypothesis, the anticodon to codon interaction will come. Now you see here, this is the CAGU is the nucleotide sequence in the codon. Suppose this is a codon, this is a nucleotide sequence in the codon. Now when we use the nucleotides in the anticodons, we observe that instead of C, what should come? G, instead of A, it should come U, instead of G, it should come, complementary to that sequence will come. These are usual nucleotide codons which should be present. But in wobbling, what happens? Instead of G, if I is present, that will also go and bind with C. If instead of U, if you have I, that will also go and bind with adenine. Instead of C, if you have U, that will also go and bind with guanine. So this is what the Vowels hypothesis, Vowels nucleotide means. Usually the vowel position is at the 5' end of the anticodon arm. That means 3' end of the mRNA. This particular nucleotide, there will be degeneracy. It keeps changing, but the sequence of, the tRNA will be read properly. In case of eukaryotes, you just see, you have a vowel codon as U, here, this place. In U, if the possible anticodons can be guanine or inosine. If you have C, the possible anticodons will be again guanine or inosine. If you have adenine, the possible anticodons will be uracil. In case you have guanine, the possible anticodons will be cytosine. So is it clear, this one? Usually what should happen? If you have uracil, it should have what happen? What will be present here? It should be guanine, cytosine, guanine, adenine, uracil, guanine, cytosine. But you have additional base which is coming here, nucleotide, this inosine. So instead of U, it should come I. Instead of cytosine, you can get inosine. So this happens at this 5' end of the anticodon loop, or as the last sequence of mRNA, that is the 3' end. This place there will be wobbling. Okay? So this is what is important. Now second last is unambiguous. Without any doubtful meaning, only one codon stands for only one amino acid. That's what I told. That means I told you, C1, C2, C3, these are different codons, they will code for one amino acid. 61 codons will code for 21 amino acid. Okay? The last is universal. Universal means the codons are same for all species on earth. It is not that the sequence of codons will alter with the different species. Now if you see here, elephant and E. coli, from elephant to E. coli, all the codons will be same. Okay? So termination codon UAG and UGA. Initiator codon will be AUG, which is a initiator codons. Okay? So these are the exceptions of genetic code. You have AUA. In cytoplasm is translated into isoleucine, and in mitochondria this is translated into methionine. These are the exceptions. UGA, which is the termination codon, but this will code in the mitochondria as tryptophan. AGA, which is coding for arginine in cytoplasm, but in mitochondria it codes as a termination codon. Similarly, AGG is an arginine coding in the cytoplasm, but it will be a termination codon in the mitochondria. So these are the different exceptions for the genetic code. So today we have discussed about the tRNA, the salient features of the genetic codes. Okay, we'll recap this one. We were talking about the DNA replication, then we talk about transcription, and then we moved on to translation. Is ultimately reading the sequence of the mRNA to form a protein. Okay, we discussed that when the mRNA sequence are formed, this mRNA sequence will be that equal to that of non-template strand, because it is coding just opposite of template strand from 5' to 3' end, and these will be coded, this will be read in the form of codons, triplet codons, and which will be alternatively forming different amino acid and giving rise to proteins. Okay. For protein synthesis, the translation process, you require tRNA, you require mRNA and the genetic code. You also require ribosome, that is 30S subunit, 50S subunit. I will discuss about those. Then the translation process per se as such. Okay. Now, this tRNA we discussed about the tRNA, the silent features of tRNA. You have two different arms, that is D arm and the T arm. Okay. And I discussed about CC arm, which is nothing but the long arm where the attachment of tRNA is placed, and this will be determined by the sequence of anticodon loops which are present in the tRNA. Okay. So, this is important. Then I discussed about the different codons coding for the same amino acid. This is extremely important. As you see, arginine coding 6. Similarly, you have tryptophan being coded by only one codon. So, there is something called degeneracy. That means 61 codons which are present, they will code only for 20 amino acids. So, there comes the degeneracy. Okay. Then these are the features I discussed: triplet codons, universal, degeneracy, unambiguous, unambiguous, then non-overlapping and non-punctuation. Okay. And these are the different characteristic features I discussed about the codons and the Vowels hypothesis. So, with this we wind up is today's lecture. In the next lecture we will continue further about translation process. Thank you.